Hello friends, BioVerse പുതിയ വീഡിയോയിലേക്ക് ഏവർക്കും സ്വാഗതം. കഴിഞ്ഞ വീഡിയോയിൽ നമ്മൾ DNA സ്ട്രക്ചറും RNA സ്ട്രക്ചറും അതിൽ ഇമ്പോർട്ടൻ്റ് ആയിട്ടുള്ള കുറച്ച് ടെർമിനോളജീസും ചോദിക്കാൻ ചാൻസ് ഉള്ള കുറച്ച് ചോദ്യങ്ങളുമായിരുന്നു നമ്മൾ നോക്കിയിരുന്നത്. ഈ വീഡിയോയിൽ നമ്മൾ DNA as genetic material, the evidence for this ആണ് നമ്മൾ പഠിക്കാൻ പോകുന്നത്. എന്ത് എവിഡൻസ് വഴിയാണ് DNA ആണ് ജനറ്റിക് മെറ്റീരിയൽ എന്ന് കണ്ടെത്തിയെന്നുള്ളതാണ് നമ്മളിവിടെ നോക്കാൻ പോകുന്നത്. അപ്പം DNA ജനറ്റിക് മെറ്റീരിയൽ അല്ലെങ്കിൽ ഹെറിഡിറ്ററി മെറ്റീരിയൽ ഇസ് ദെയർ എന്നുള്ളത് ഒരുപാട് മുന്നേ തന്നെ മെൻഡലും ഗ്രിഗർ മെൻഡലും പിന്നെ അതുപോലെ മോർഗനും ഒക്കെ പറഞ്ഞിട്ടുണ്ടായിരുന്നു ഇൻഹെറിറ്റൻസിന്റെ കാര്യം, ജീൻസിന്റെ കാര്യമൊക്കെ. പക്ഷെ ഈ ജീൻസ് അല്ലെങ്കിൽ ജീൻസ് ഈ ക്രോമോസോംസിന്റെ കെമിക്കൽ നേച്ചർ എന്താണ്, അല്ലെങ്കിൽ ജനറ്റിക് മെറ്റീരിയലിന്റെ കെമിക്കൽ നേച്ചർ എന്താണ്, അത് DNA ആണോ, പ്രോട്ടീൻ ആണോ, അതല്ലെങ്കിൽ കാർബോഹൈഡ്രേറ്റ്സ് ആണോ, ലിപ്പിഡ്സ് ആണോ എന്നുള്ള കാര്യങ്ങൾ അറിയാനായിട്ട് ഒരുപാട് ടൈം എടുത്തു. ഓക്കെ, അപ്പം അതിനിടയിൽ വന്ന ഒരുപാട് കുറെ എക്സ്പെരിമെന്റ്സ് ആണ് ശരിക്കും സീക്വൻഷ്യലി ഈ ഒരു DNA ആണ് ശരിക്കുമുള്ള ജനറ്റിക് മെറ്റീരിയൽ എന്നുള്ള ഒരു കൺക്ലൂഷനിലേക്ക് എത്തിച്ചത്. അപ്പം ആ കുറെ എക്സ്പെരിമെന്റ്സ് ആയിരിക്കും നമ്മൾ ഇന്നിവിടെ മെയിനായിട്ട് ഫോക്കസ് ചെയ്ത് സംസാരിക്കാൻ പോകുന്നത്. So the first experiment is the Griffiths Experiment on bacteria called Diplococcus pneumonia. Diplococcus pneumonia is of two strains. One is the virulent strain and second one is the non-virulent strain. Virulent strain is also known as smooth strain because it contains, or S strain, which contains a smooth coating of mucus over its cell wall, while rough strain or R strain is its cell surface, its cell wall is kind of rough because of the absence of this mucus over its cell membrane. And because of this rough nature, it is identified by the immune cells easily, and this is non-virulent in nature, while the smooth strain, which contains a smooth coating, escapes or evades the immunity, and it can cause pneumonia in the infected person. Okay, so there are two strains of Diplococcus pneumonia: virulent or S strain or smooth strain, which contains mucus coat over to produce a shiny colonies, and it causes pneumonia. It is virulent and it causes pneumonia. And the second strain is non-virulent or R strain or the rough strain, which is non-capsulated, though there is no mucus coat, and thus does not cause any pneumonia. What he did was, he first took mice and he infected the mice with the rough strain or the non-virulent strain. So, as obvious, because the rough strain is non-virulent in nature, the mouse survived, or the mouse lives. Okay, and in the second set of experiment, he infected the mice with the smooth strain or the virulent strain, and the result was, here also the result was obvious, because the virulent strain can cause pneumonia to the mouse. It dies. Okay, next in the next row, what he did was, he treated the smooth virulent strain. He treated he killed the smooth strains. Okay, so because the strains were dead, it was unable to infect the mouse, and the mouse lives. There was no pneumonia in the heat killed smooth strains. In the next set of group, what he did was, he took the heat killed strain and the rough strain. He mixed both the bacteria together and then he infected the mice. But surprisingly, the mouse dies. Okay, the rough type of bacteria and the smooth type heat killed bacteria were injected together in the mice, and the mice died due to pneumonia. Not only this, he also recovered live smooth cells from the smooth bacterial cells from the dead mouse in the fourth group. Okay, so from this he concluded that occurrence of living S-type virulent bacteria is possible only by their transformation from R-type. So he said that the live R-type bacteria which were infected to the fourth group in the presence of the dead S-type bacteria, it got transformed into the S-type bacteria to cause pneumonia. So something from the S-type bacteria was getting into the R-type bacteria to convert it into a new strain or S-type strain that can cause pneumonia, and this is known as Griffith's effect or transformation. Okay, but here Griffith's experiment cannot prove following points: whether the transformation was only possible if the mouse was there. If mouse is absent, whether the transformation and will not occur. This was not proved. Okay, so in the absence of mice, also the transformation is occurring or not was not proved here. The second thing was whatever the transforming principle was, whatever what is getting converting the R type to the S type, was it the polysaccharide in the mucilage or the protein or the DNA in from the S bacteria? It was unknown from this experiment. Griffith's experiment fails to address the chemical nature of the genetic material. But in 1940s, കുറച്ച് മൂന്ന് സയൻറ്റിസ്റ്റുമാർ, Avery, Macleod and Carty, they did an experiment inspired by Griffith's experiment only. Okay, what they did was they took the same heat-killed smooth strains. The same kind of experiment was set there, but this time they took the heat-killed S cells. Okay, what they did was they heat-killed the S strain, then they removed the lipids and carbohydrates from the sample. Okay, then the remaining cells or the sample contained proteins, RNA and DNA. Okay, then they subjected the solution to different enzymes. One was treated with proteinases, second was treated with the ribonucleases, and third was treated with deoxyribonucleases. Proteinases, as we know that it degrades the protein, so the sample contains no protein. Ribonucleases degrades the RNA, so the sample will not be having any RNA in it, and the deoxyribonucleases degrades the DNA, so the third sample will not have any DNA. So the experiment was set to identify the chemical nature of the transforming principle. The transformation is occurring, so something was transferring from the S strain to the R strain. So what they did in the next step was they took the R strains and they mixed it with the heat killed S cells as Griffith did. Okay, but this time this was set in three different experiments or three different groups. Okay, one containing proteinases that had no protein. So if the proteins are the transforming principle, there should not be any transformation. The second group was added with ribonucleases. This sample contained no RNA. So if the RNA is the transforming material, this set of group will not be showing any transformation. And the third group was treated with deoxyribonucleases. So there will be no DNA, and if the DNA is a transforming material, it should not be forming any transformation. Okay. So when the colonies were checked for further for transformation, Avery, Macleod and Carty found that the samples containing proteinases and the RNases got transformed, while the sample containing deoxyribonucleases, where DNA was not present, there was no transformation. That means in the absence of DNA, there was no transformation. That means the transforming principle is the DNA. Transformation was occurring because of the DNA. Okay.